Hello, good evening. Uh, this is your third week session with me. So, in this week, we'll be talking about the third week topics, which were homologous and non homologous recombination and site specific recombination, and then we'll practice MCQs. We'll also look into the current literature, and then I'll be taking your doubts regarding this session. Okay, so first of all, I'll give a brief recap of the previous session. So in that we looked into the DNA repair and how the DNA get damaged. So in which we looked that DNA is constantly exposed to different damaging agents, which could either be exogenous or endogenous, and this damage further leads to mutations, and these mutations ultimately leads to genomic instability and which cause different diseases. And if this DNA is not repaired, then your cell will ultimately leads to cell death. So that's why we need repair mechanisms which ensures that this genetic information is preserved and your cell do not die. Without repair, uh, there are chances that you can get cancer, premature aging, or cell death. And then we also looked into the type of DNA damages, in which we looked into endogenous sources as well as exogenous sources. So endogenous sources means which are within our body. So for example, reactive oxygen species which are formed due to the result of metabolic processes. Then replication errors and mismatches. So our replication machinery is not 100% accurate. There are chances that there might be some errors. Then topoisomerase DNA complexes. So this topoisomerase is very important to relieve the stress which happens during the helicase unwinding of DNA. So in that case, if topoisomerase do not work properly, then there are chances that your DNA get damaged. There could be double strand or single strand breaks. Then comes exogenous sources. That means some external agents such as UV radiations, which leads to the formation of thymine dimers, ionizing radiations such as X-rays, gamma rays. So if you are frequently exposed to these radiations, then there are chances that your DNA might get damaged, but 1 or 2 second or means lesser time interval exposure is not that harmful. Then comes chemical mutagens and viruses. So these are also exogenous sources that can damage your DNA. Then we looked into the DNA repair pathways, in which we discussed in detail about the base excision repair, which is used generally when we have small single base pair faults or some mismatches. So in that case we use base excision repair, and which is mainly used. Then comes nucleotide excision repair. It is used when we have bulky DNA lessons or if we have more than one base pair damage. Then comes mismatch repair, and it generally is used when there is mismatch in your DNA sequence. And this mismatch repair mechanism, it's very expensive. It requires a lot of energy and it's quite slow process. So it is not much favored. Then comes homologous recombination, which has high fidelity for double strand break repairs. So the above ones which we discussed in previous session, they were about one or the smaller fragment damages. But if there is double strand break, single strand break, then we generally go for homologous recombination, and this is for if we want to treat the larger base pair damage. And then comes non-homologous end joining. So in today's session we'll look into the detail of these recombination processes. So in homologous recombination we have the homologous strand from where we'll take the information, whereas if we talk about non-homologous end joining, it simply means if there is some break, you simply join them. So the information which is deleted, that remains deleted. So that's why this is quick, but it is error prone as compared to homologous recombination. And then another one is alternative end joining. So this is nearly similar to NHEJ, but it is a backup mechanism. So it is only favored if the non-homologous end joining is not working properly. And then direct reversal. So this was the first DNA repair pathway which we discussed. So in this case enzymes are used and they directly means treat the damaged DNA without any synthesis process. So it's very quick. So this is all which we have discussed in our previous session. So in today's session we'll be talking about the double strand break repair, or the bigger damages that happens to the DNA. So before moving further, I'll just check if isn't okay. Yeah, so in double strand breaks, these are the most lethal DNA damage types, and it can happen due to ionizing radiations, or if there is replication fork collapse, or if there is oxidative stress. And if it is not repaired, then it can lead to chromosomal rearrangements and loss of genetic information. So importance of this double strand repair means double strand break repair is that to ensure that the cell survives and regardless of how the damage has occurred. So in this we will be mainly focusing on three main mechanisms. One is homologous recombination, second one is non-homologous end joining, and third one is alternative NHEJ or alternative EJ is also another name for this. So in homologous recombination, as the name suggests, homologous, which means we are using some homologous information. So homologous means if two sequences share some kind of similarity. So in this case we use intact sister chromatid as a template. So we are using some template to take information from that template and synthesize the region which is broken in the DNA which is being considered. And it has high fidelity because we do not lose any information, and the enzymes which are used are quite quick, the polymerases which are used. So this has high fidelity repair because there are lesser chances of mismatches or any other things. It retains all the genetic information at the break site. And if we talk about NHEJ, which directly rejoins the part which is broken. So in this case, if we have lost some base pairs during the damage, then we'll not be able to get them back. So in this way there is the loss of information at the break points. That's why it is also called error prone, because it might introduce small deletions at the break site. And then comes alternative NHEJ, which is alternative form of non-homologous end joining. It is quite slow and it is mutagenic as well, so there are chances that there might be some mutations or some changes, and it is more error prone than the NHEJ. So in this case also, if for example if there is double strand DNA damage, then we might lose the information. In this case we are not losing any information after the DNA is being broken, but in case of alternative end joining, there are chances that we might lose some other base pairs also while joining both the DNA segments, and there might be some mismatches or mutations that can also take place during the alternative end joining. Okay, so this is the overview of different enzymes which are being used in each mechanism, and means this slide is like where complete information is being kept, but in later slides one by one we'll go through each process. So for example, if we talk about homologous recombination, so in this case different enzyme complexes come and they bind to the site where the DNA damage has happened. So most likely it is the mRN complex, which is the complex of three different enzymes, and CTIP. So this is also another enzymes which bind to this region and they start the resection process in which they degrades the bases which are present at that break point. So DNA ends are resected by nucleases. So nucleases are the enzymes which degrades the DNA, and generally this degradation takes place from 5' end to 3' as well as 3' to 5' end at this break site. And then this single stranded DNA, it is coated with another protein which is RPA, which stabilizes this when this degradation is happening. So it stabilizes the single stranded DNA. Then there is homology search, because in homologous recombination we need to find the sister chromatid which will further act as a template, and then we can construct the new DNA fragment to that. So this homology is being searched and then precisely the break is being repaired. So there are other enzymes also which takes place which comes. So different proteins also help this, different cofactors are involved. So in this slide we'll not go into the detail of each cofactor, but this is just an overview that how generally the process happens. And then if we talk about non-homologous end joining, then in this case the difference from HR is that in place of mRN complex there is Ku7080 recognition complex. So these two proteins, they recognize that where the double strand break is present, they bind to that region and then they further recruit DNA PKs. So further protein kinases come which phosphorylate those sites, different chemical processes takes place. Now after this phosphorylation has been happened, then it acts as a signal for other proteins so that they can recognize the site where the damage has happened. So further other factors comes. So these factors are the proteins or enzymes which are involved in this non-homologous end joining pathway. So these are some of the enzymes which comes, such as XRCC4, then XLF, then ligases 4. So different enzymes comes, and they then start joining the where the damage has happened. So like 4 ligase 4 is the enzyme which is used to ligate it, and then these other protein complexes, they also support so that the ligase can join these fragments. So ultimately we have ligated the 2 DNA strands where the break has happened. Then comes alternative EJ, which is alternative end joining. So this is for 2 to 20 base pair damage. And then SSA, which is the single strand annealing. So this is for greater than 25 base pairs. So in this case, mRNA and CTIP, same as HR, these proteins come, they recognize the damaged site, and then they start the resection process. Then alignment of homologous sequences. So in this case, they both rely on the short homologous sequences which are revealed by resection. So when the double strand break has happened, so these breaks, so in this case you can see this is like a blunt end, but in the case of alternative end joining, the break is like there is some flanking region which is present. So this flanking region, it can act as the template for the above strand, so that it can synthesize the nucleotide which are complementary to the bottom part. So in this way it is relying on that short homologous sequences which is revealed by resection. So after that also it will further resect towards the 3' end and the 5' end. So more of the flanking region will be exposed, which can act as the template. Now after that it aligns the sequences and it removes the flaps. So if extra sequence is present, then it will be like a flap and it will be ultimately removed, and the 2 DNA fragments, they will be joined. So for example, this break point, this will be joined by the other set of ligases. So in this case also ligases will come. So it uses 2 to 20 base pair homology. That means if the damage has happened, so it will cleave up to 2 to 20 base pairs, and then the bottom part will act as the homologous template for it, or sister chromatid for it, and then synthesis will take place. So if there is only single strand break, then it can anneal up to 25 or more than 25 base pairs. Okay, now we'll start with homologous recombination. Now in this we'll look into the detail of all the enzymes and proteins which are used and what happens in the case of this homologous recombination. So the purpose is that to accurately repair the DNA double strand breaks using intact sister chromatid as a template. This has high fidelity. So why high fidelity? Because no information is getting lost, because we have intact sister chromatid. So wherever the break has happened, we know that which sequence or how many base pairs are missing, so we can construct those base pairs. And it works mainly in the S and G2 phases when the sister chromatids are available. Now the key players or key proteins which are used in this process are mRNA complex, which we discussed. So mRNA complex is made up of 3 proteins, which is MRE11, RAD50, NBS1. They senses the double strand breaks and start the processing of the ends. Then BRCA1 and 53BP1, so it decides that which mechanism to use to cure this double strand break, either to use homologous recombination or should we use non-homologous end joining. So it depends that in which phase your cell is. So if there is no sister chromatid available, then we can not go for homologous recombination. Then comes RPA. So this is another protein which binds to single stranded DNA and protect it from the nucleases or other degrading processes. And also because DNA is unstable when it is single stranded, it tries to re-anneal. So to make it stable in that single stranded form, RPA is required. Then comes RAD51, which will further replace this RPA, and now it will perform homology search and strand invasion. So it will search for the sister chromatid and then it will invade it. So in the next slide I will show you that how it invade, so how the holiday complex and all those complexes are formed, so that it can act as the template. Then comes BRCA2 and PAL-B2, which helps to load this RAD51. Then there are some other accessory proteins which are like additional proteins, not directly involved, but they have some other roles, such as RAD54, RAD51 paralogs, BLM helicase, then resolvase, such as MUS81 Gen 1, SLX1. Now the question is that why this homologous recombination is important. So we've already discussed, it is error prone, it prevents mutations, because in case of alternative end joining there are chances of mutations, but in this case there are no chance of mutation. Then it is also essential for meiosis and generating the genetic diversity, because in recombination we know that two different uh, two different chromosomes, they share some genetic components, so in this way they can generate genetic diversity. Okay, so key decisions in the repair of double strand breaks by homologous recombination. So first decision is that the DNA ends, they are bound by either mRN or Ku70-AT. Now this protein complex will decide whether it will undergo homologous recombination or non-homologous end joining. So Ku70-AT are used in non-homologous end joining, whereas mRN is used in case of the homologous recombination. So first decision comes after the binding of these protein complexes. Then the second decision is short range resection by mRN. So there are chances that how much damage has happened. Is it short range, or how much resection need to be performed, short range or long range. So that decision is made by mRN. Then long range resection of the DNA and RPA binding. So then third decision is, if there is long range resection, then after the long range, means when the enough resection has happened, then there need to be binding of RPA, which is necessary to stabilize the single strand, because we have degraded the bottom strand. So then RPA will come. This is our third decision. And fourth is that this RPA get replaced with RAD51. So this replacement is also known as RPA to RAD51 switch. So it is exchanged. This facilitates the search of the sister chromatid or homologous part, and then it will also lead to strand invasion and DNA synthesis, which is necessary for homologous recombination. So these are the decisions which are taken during homologous recombination process, that when to use which protein complex. Now these are the steps. So first step is that whenever the damage has happened, then we need to recognize where the damaged place is present. So to recognize it, the proteins are the mRN complex. So these are three proteins which make complex, and they search the entire double-stranded DNA to find the site of, to find the break point. And after they have identified, then they recruits ATM kinases to activate the DNA damage response. So ATM kinases, they act as a signal, because they will phosphorylate that part and they will send the signal that this part need to be repaired. So in this way, this is known as the recognition of damage and initiation of the process of DNA damage repair. Then comes end resection, in which these ends, they are processed. This means they are cleaved to generate the single stranded DNA part, so that we can start the new repaired DNA from there. So we can synthesize a new strand. So this processing takes place. So firstly, there is short 5' to 3' trimming which takes place, in which mRN complex and this CTIP, these proteins helps. Then extended resection of this means this extended resection is favored by the nucleases enzymes, which generates the 3' single stranded DNA tails, because now it is long range resection. That means greater part is being deleted. Then RPA will coat the single stranded DNA so that we can prevent its degradation. So here you can see that after the long range resection has happened, so RPA comes and it binds to that single stranded part, so that other nucleases do not degrade the bottom strand. For example, if we talk about this particular strand, then topmost strand is resected, but we don't want that here also resection happens. So RPA will protect that end. Now after long range resection has happened, so now this will send the signal to the machinery that okay, enough damage has happened, now RAD51 will be loaded, RPA will be replaced. So now in this case RPA is replaced with RAD51, it will bind. Now RAD51's work is to find the sister chromatid or the homologous region. So RAD51 will search for that. Now BRCA2 and PAL-B2, they replace the RPA with RAD51. So these are the other proteins which are involved in this replacement. So as here you can see PAL-B2 and BRCA2. So remember these names as well. So they help the RAD51 to replace the RPA to form the presynaptic filament. This particular filament is known as presynaptic filament. And then synapse is the point where the crossing over happens. So if you have read about meiosis in that way. So in that we talk about the presynaptic complex and all those things. So here also this particular complex which is being formed, this is presynaptic filament. This will later on search for the homologous part or homologous sister chromatid, and then strand invasion will take place, which will be helped by the RAD51 only. So RAD51 filament, it will invade the homologous DNA duplex. It will create a D loop. So this kind of loop is known as D loop or the displacement loop. Now why this D loop is required? Because if you see this topmost DNA fragment, it requires some template. So for this, D loop will now act as the template for the polymerases to synthesize the new DNA fragment here. And same goes for this particular part. So here also D-like structure is formed. So this is known as D loop. And this D loop is also created by RAD51 when it is helping the DNA to invade the homologous stretch. Now DNA synthesis will take place, in which DNA polymerases are used, and specifically when the repair mechanisms takes place. So polymerase delta is used in case of eukaryotes. They copy the DNA from the intact template. Then helicases are required like HROB and MCM89 and HelQ. So these helps to unwind the DNA for synthesis. Now formation of joint molecules. So after this, when the synthesis will take place, so the formation of joint molecules will be there, which later need to be separated. So now different complexes could be formed. One is SDSA, which is synthesis dependent strand annealing, in which there is no crossovers which are formed. Then comes BIR, which is break induced replication, which is long range DNA synthesis. And the third one is double holiday junction, which can lead to crossovers, or it can either form non crossovers as well. So in the later slides we'll see them one by one, but this means how your joint molecule will appear, whether it will lead to crossing over or non crossover. So these are the structural representation, how they appears and what will be the outcome. And then similarly we talk about resolution. Then dissolution is BLM top 3A RM1 RMI 1/2 complex. They separate these junctions without cutting them. And endonucleases comes, which are MUS81, EME1, Gen1, SLX1. They cut the DNA to complete the repair process. So ultimately you will get the error-free repair. So in the end we get double-stranded DNA, and these are different proteins which facilitate this. Okay, now one by one we'll talk about all the three joint molecules which can be observed. So during the repair mechanisms there are different paths which repair mechanism can take. So different decisions are taken during this process. So after the strand invasion takes place by RAD51 coated single stranded DNA, D loop is formed, which we discussed, which is like a displacement loop. Now after the synthesis takes place, then there is possibility that three types of joint molecules can be synthesized. So first one is SDSA, which means synthesis dependent strand annealing. So in this case the invading strand primes the DNA synthesis using a homologous template. So in this case, when this particular invasion has happened, so this fragment will synthesize the complementary DNA to our invaded strand, and newly synthesized strand will be displaced and annealed to the other break end. So in this way, this represents, the red color dot represents the newly synthesized DNA, which is complementary to our sister chromatid. So in both the parts, this new DNA has been synthesized complementary to our sister chromatid. So no crossing over has happened in this case. Advantages is that it preserves the genome structure, and it is used for most mitotic homologous recombination events. And one example is of yeast mating type switching. So whenever yeast mates, so they also perform this type of sharing of genetic material. Now second is classical DSBR, which is the double holiday junction, which is most commonly observed. So this formed when both the ends of this break engage the homologous template. So in this case both the ends are engaged. DNA synthesis from both the ends plus ligation creates two linked DNA molecules. So in the case of SDSA, in this case single strand was getting annealed one by one, whereas in this case when both the strands are involved at the same time, so this particular complex, this junction which is formed, this is known as double holiday junction, and the resolution options are okay, somebody has yeah, so in this case of double holiday junctions, both the both the broken ends, they engage in the homologous template synthesis, in which the structure of double holiday junction like structure is formed, and nicking and ligation. So during nicking and ligation, crossing over or non-crossover products can be synthesized depending on how the cleavage takes place. So resolution options, okay, so it depends that if how the cleavage is taking place. So if cleavage is not appropriate, then it can leads to crossovers, and if the cleavage is properly taking place, then it will be the new DNA strands. For example, if there is no cleavage and this particular fragment, the second strand, it goes up and this strand comes down, then it will be non-crossover. But if cleavage takes place over here, then these two strands, strand 2 and 3, they will be the recombined, or you can say the variant strands which are formed. So in this way, there are chances that you can, in the result, you can either get non-crossovers or crossovers depending on how the cleavage is taking place. So if dissolution is taking place by BLM top 3 alpha RMI 1 by 2 complex, then no crossovers will be formed. So it will simply dissolve it. It will simply remove the joint fragment which are synthesized. But if there is nicking, then it will lead to the generation of crossovers. So key point of this double holiday junction is that the crossovers are common in meiosis, but it is rare in mitosis to avoid the chromosomal rearrangements. Okay, in this case, BIR, which is the break-induced replication, so it occurs when only one end of double strand break find the homology. For example, this collapsed replication fork structure. In this, strand invasion initiates the long-range DNA synthesis, and it can also copy the entire chromosome arm. This is conservative DNA replication, both the new strands made from the invaded template, but only because just one strand is invading the complementary sister chromatid, so it can recover, but the other strand, it do not have any complementary sister chromatid, so due to which no proper annealing is taking place. So that's why it is known as break induced replication. This is conservative DNA replication, both the new strands made from the invaded template, and the biological role is repairing the broken replication forks or telomere maintenance in the alternative lengthening of telomere pathways. But the risk is that it can cause the loss of heterozygosity, because the region which is lost, it cannot be recovered. So ultimately this particular strand which is getting synthesized, because in one chromosome we have these two different sister chromatids, and for this particular chromosome we have only one homologous sister chromatid. So for the other chromosome we do not have, for the other chromatid we do not have complementary or homologous sister chromatids. So due to which to fully construct it, we will be using the same information for the same sister chromatid which we have used for this second sister chromatid of this case, second chromatid of our damaged DNA, due to which we will lose the information which was present in this. So heterozygosity will be lost, and it will be more like a homologous chromatid. So maybe in later slides it will be more clear, but the main mechanism on which you should focus is this double holiday junction, because this is majorly favored, and then SDSA is also favored. BIR is very less, it's not used, and it takes place only if both these mechanisms fails, if both these repair mechanism cannot take place. Now we will look at the molecular mechanisms of repair, which is REC-BCD pathway. So REC-BCD this is present in bacteria. So these are the prokaryotic enzymes which are present. So it is a multifunctional enzyme complex in bacteria. It functions as a sequence regulated bipolar helicase nuclease, and its role is to process the DNA ends which results from the double strand breaks. So this is the structure of rec-BCD. So in this case, the rec-B, it has the helicase activity and nuclease and rec-A loading activity. Rec-D also has helicase activity, and rec-C is the region which has the site which can bind to the chi site. So first of all, rec-C, it recognizes the chi site, which is this sequence, and then rec-C, it will send the signal to rec-D. It will tell the rec-D to stop, and when stopped, this rec-D signals rec-B nuclease to swing. Then this rec-B will move towards this end and it will cut where it is. So rec-B continues the unwinding of DNA, and ultimately it will load rec-A. Yeah, so one by one when we'll move, then this will be more clear. So mechanisms is, firstly, whenever there is double strand break, so binding of this rec-BCD will take place to the double strand break. DNA strand will start to unwind and it will synthesize the single strands. Then degrading of single strands will take place with the help of nuclease. So these complexes, they also have different proteins are used which have the nuclease activity, which will degrade the double stranded DNA, which will synthesize the single strands. Now degradation will stop when the chi site is obtained or chi site is reached, so which act as a recombination hotspot. So this chi site is also known as recombination hotspot because this is the site where the recombination more likely takes place. The frequency of recombination is high. So imagine that this particular protein complex, it is binding to the site of damage. Now what it will do, it will, for example, if the blunt end cut is there, so it will recognize that site, it will bind to it, and it will start unwinding the DNA, constructing two different single strands. Then with its helicase activity, it is doing unwinding. Then it will call the nucleases which will degrade the single strand. So one single strand will be degraded, and this process will take place till the chi site is reached. When this rec-C will identify the chi site, then it will send the signal to rec-D part that stop the unwinding, stop this process. So at that point, the process of the degradation will stop, nuclease activity will stop, and rec-A will be loaded onto that 3' single stranded DNA tail. So now what are these chi sites? So they are short DNA sequences which will stimulate the homologous recombination, because we have seen in the previous slide also that how when RAD51 comes, then it search for the homologous chromatids. So similarly in this case also, as soon as chi site is reached, then it will call rec-A, and then some other proteins will come which will start to search for the other complementary sister chromatids so that recombination can take place. So in this way it acts as a recombination hotspot, and this sequence is species specific within the related bacterial groups. So in E. coli and Salmonella, the core chi sequence is this one, which is composed majorly of GGG and T. And this additional 4 to 7 nucleotides on the 3' side of the core are important for its activity. Okay, then comes rec-A proteins. So here it was rec-BCD, and then we said that in the end, after chi site has been reached, it will call the rec-A. So rec-A, this is the length of rec-A protein, this is the mass, and it is conserved in bacteria. It is also observed in chloroplast, archaea and eukaryotes. So function of this protein is to bring about homologous recombination and SOS response. So SOS is like save our soul response, which is the last DNA repair process, takes place in prokaryotes when no other mechanism works. Then SOS is the end goal, means when nothing is left, then SOS repair mechanism get activated, and in which different enzymes are used. So in that case, rec-A is also used in this pathway. This is ATP driven homologous pairing and strand exchange pathway, ATP hydrolysis with single strand DNA, and ATP dependent single strand DNA uptake and hybridization. So these are the functions of rec-A. But keep one thing in mind, that this mechanism, it is ATP driven. That means it requires energy. So this is a single stranded DNA which is synthesized. Then this particular part represents the rec A and green dot represents the ATP. So it will bind to the single stranded DNA and it will further attach to the ATP. Then ultimately it will send signal to other rec A which is present, and ultimately it will assemble all the rec A on the single stranded DNA filament. When different rec A will come, at a point will come when the rec A and single stranded DNA filament, there will be saturation, that all the bases have been covered by the rec A protein along with ATP. So mechanism is same what we have discussed. So single strand DNA, it will bind to site 1, which is the nucleoprotein filament, forms 6 monomer per turn. ATP activates the filament. So this site 1, it is present in the rec A protein. Rec A protein is very long, different sites are present. So similarly they are talking about the site 1. So this single stranded DNA binds to that particular site. Then ATP will come, which will activate this filament. Homology search will take place, in which double stranded DNA will bind to this N terminal domain, and it will further move towards the C terminal domain, which is further site 2 will be present. So if homologous, then strand exchange will take place, and if there is no homologous DNA present, then it will be releasing. So it will release this rec A and ATP, everything this will release. Okay, so now this is the molecular structure of rec A protein, that how it looks. So in this is the ribbon representation and this is the surface representation. So this rec A protein is very big. It has N terminal domain towards its N site. Then it has site 1, site 2. One site is ATP binding site. When the ATP binds, then this protein get activated and it will further call other rec A proteins to assemble on the single stranded DNA. It has some catalytic sites or some residues which are present, such as this glutamine and glutamate at 96th position. It has C terminal domain, and then this lysine arginine motif. So in this same color represents the same surface, so it represents what kind of interaction is taking place. So in this red surface represents where the double strand DNA interaction takes place, whereas this orange part represents where the single strand DNA binding takes place. This particular region is involved in the hydrolytic activity. This 2 represents the ATP binding site. Similarly, this 5 is also involved in double strand DNA binding. Now this 5 and first, which is in orange, so these 2 regions, they are involved in homology search and strand exchange. And similar thing is defined over here. Okay, so before moving to eukaryotes, we'll again go through this rec BCD and rec A. So as we discussed in this particular case, so here we talked about mRN, then we talked about RPA, and then RAD51. So similarly in prokaryotes, instead of this mRN1, we discussed that how this rec BCD will come. So rec BCD will come, it will bind to the site where the break has happened, and then after binding to this site, because this rec BCD, it has different components B, C, D, they have helicase activity, in which firstly they will unwind this double stranded DNA, then with their nuclease activity they will degrade one of the strand, so that degradation will take place, and this degradation will continue till the point when the chi site is reached. So chi site is the stretch of sequence which has the complementary region present in the rec C. So rec C, whenever that chi site is reached, then it will send the signal, it will say that stop the nuclease activity. So at that point nuclease activity will stop. Now rec A will come. So as in this case we had RPA and RAD51. So similarly in this prokaryotic case we have rec A. So rec A will come, it will bind to the single stranded fragment which is achieved. So then it will look for the complementary sister chromatid or homologous sister chromatid, so that the recombination process can take place. So these are the proteins which are involved in the bacterial system. Now we will talk about homologous recombination in the eukaryotes. So first step is resection, in which the recognition of double strand break is done. So in this case mRNA comes, which we have discussed. These 3 protein complex, it detects the double strand breaks. They will recruit ATM kinases, which will phosphorylate this site, which will send the signal that the repair need to be done here. Now BRCA1 versus 53BP1, they have opposing roles. They will decide that whether the repair will be homologous recombination or NHEJ. So BRCA1, it initiates the resection and it helps to load RAD51. So after the RAD51 loading takes place, it will search for the homologous histochromatid and then strand invasion will take place. So this resection which is taking place here with the RPA, so RPA is stabilizing this, but this resection which is taking place, so this resection creates 3' single stranded DNA which is coated by RPA for protection. Then comes BRCA2 and PAL-B2 complex, which will help to replace this RPA with RAD51. Now it will form RAD51 and single stranded DNA presynaptic filament. This filament will scan the genome for the homologous double stranded DNA. So RAD51, keep this protein in mind, that RAD51 is the protein which is involved to search for the homologous double strand DNA. And upon finding homology, it will do the strand invasion and D loop will be formed. So after the D loop has been synthesized, now the 3' invading strand, it will prime the DNA synthesis, and then key polymerase here is polymerase delta, which will synthesize the complementary strand. So after this D loop has been formed, complementary strand need to be synthesized, which will be done by DNA polymerase delta. The helicases which will come are HROB, MCM8 and MCM9 and HELQ, which will promote this synthesis, because we know that during the DNA synthesis we require the unwinding of double stranded DNA. So this unwinding of the sister chromatid which has been used as a template, so for that unwinding we require these helicases. Now next step is the HR intermediates and resolution. So we discussed that 3 different ways in which the double strand break can be treated. So one is the construction of double holiday junction pathway, and other one is the synthesis dependent strand annealing, which will means always generate known crossovers, and then the break induced repair, in which this is for the long range synthesis, and it happens if we have only one sister chromatid which is able to find the homology, but the other sister chromatid do not have any homologous stretch. And DHJ, which is the double holiday junction pathway, which is most common, and this can also lead to generation of crossovers versus known crossovers depending on which enzymes are used. So if nicking and ligation is taking place, then it will generate crossovers, and if simply other helicases are used such as BLM, top3A, then it will generate known crossovers. So now let's talk about what is BLM helicase, which we have discussed in this case, that it will generate known crossovers. So as the name suggests, helicases. So helicases are the enzymes which are used to unwind the DNA. So it suppresses the crossovers in mitosis, because normally also this enzyme is observed in the cells. So it suppresses crossovers in mitosis. It repairs the mitotic DNA gaps by homologous recombination, and the function of this enzyme is end resection, D-loop rejection and double holiday junction resolution. And then it also has phase specific roles. So during G1 phase, it promotes 53BP1 loading, which prevents the unscheduled resection, and in S phase it promotes resection by top 3 interactions. So it depends that in which phase the cell is. So in G1 phase, where it is preventing the unscheduled resection, in S phase it will promote this resection with the help of top 3 interaction. So top 3 is another protein which is used. So in this figure, as you can see, if your BLM helicase, if it is mutated, if it is not functioning properly, then it will lead to crossing over, which can ultimately in some scenarios it could also lead to the loss of function or loss of homology, so in which genomic instability can increase and cancer predisposition can take place. So this is not always it will take place, but there are chances that if this particular BLM helicase does not work properly, if there is mutation, then this crossing over will take place and this can increase the instability. Whereas if the BLM is normal, then its role is to stabilize the genome and normal cell growth can take place. If the BLM is overexpressed, so in that case also it will lead to high DNA repair efficiency, it will lead to hyper recombination, so it can also increase the recombination process, which will be hyper recombination, and damaged DNA can also get accumulated. So hyper recombination can also ultimately increase the genomic instability, and which can also lead to tumor proliferation and chemoresistance. So the ultimate takeaway message is that BLM helicase need to be in the moderate levels, which should be normal levels, to avoid any genomic instabilities. And then ultimately comes the accessory proteins, which are the additional proteins other than these key proteins which are involved in the process. So accessory proteins in homologous recombination, they are like helper factors which do not directly catalyze strand exchange, but they assist in the co-recombination machinery. For example, RAD51 in eukaryotes, REC-A in bacteria, in carrying out the efficient and accurate repair. Similarly, we have RAD54, which is the chromatin remodeler, which will interact with this RAD51 and it will extend the joint molecules and stabilizes the D loops. Then comes RAD51 paralogs such as RAD51B, RAD51C, D. So paralogs are like the genes which arises due to duplication events. So they share some kind of similarity with this protein. So they are like duplicates of this same protein. So similarly, they are saying these are different paralogs which share the sequence similarity with our RAD51 protein. And then complexes, so BCDX2, CX3. So there are different complexes which can take place, and they all promotes the RAD51 recruitment, filament stability, and gene conversion track length. So these are talked in terms of eukaryotes, because in eukaryotes we have RAD51. Then comes SWSAP1 and SWS1 SHU complex, which is non-canonical RAD51 paralog, and it also regulates RAD51 function. So here these green oval shapes represents this SHU complex. So from this, the takeaway message is that other than all the key proteins, accessory proteins help the key proteins to be stable and perform their function properly. So in this case, the key protein is RAD51 in case of eukaryotes and REC-A in case of bacteria, which are responsible for strand invasion and recombination. So these accessory proteins help to stabilize these RAD51 and REC-A so that they can work properly. Okay, so this is the entire pathway of double strand break repair homologous recombination, which we have seen so far, that how it starts with, first of all, recognition of double strand break by mRNA, then it will recruit ATM, and then kinases, ATM kinases, which will phosphorylate that site and send the signal so that other proteins can come. So after the other proteins will come, then they will start resection of the 5' towards 3'. Single strand DNA will be available. So to stabilize that single strand DNA, RPA will come. It will bind to that single stranded DNA so that it remains stable and it do not get degraded by nucleases. And then after means when the long range resection has taken place, then another protein comes, which is RAD51, which is brought about by BRCA2 and PAB2. So they will come, they will help to replace RPA with RAD51. Yeah, so here they are bringing RAD51, they are replacing this RPA with RAD51. Now as soon as RAD51 binds, it says that now it will search for the homologous sister chromatid so that recombination can take place. So this is all about the homologous recombination. Now we will move to non-homologous end joining. So do you have any doubts till now? Okay, so if no, then I'll start with non-homologous end joining. So in non-homologous end joining also we have the canonical non-homologous end joining and the alternate non-homologous end joining. So canonical means which is the normal non-homologous end joining. So in this case, the first responder, so in case of homologous recombination, the responders were the mRNA complex, whereas in the case of NHEJ, the first responders are Ku7080 heterodimer. They will bind the free DNA ends, which is sequence independent, and they provides the docking site for DNA protein kinases, which will form DNA PK holoenzyme. They cannot bind internally blocked ends. So as soon as these Ku7080 binds, they will call DNA PKs. They will phosphorylate these sites so that other enzymes can get the signal that where the DNA repair need to be done. Now end processing enzymes such as nucleases, which are responsible for the degradation so that single strand can be generated. So nucleases will come. So in this case, the nucleases which are used are TDP1/2 or Artemis, which remove the hairpins, damaged bases and protein DNA adducts. And additional enzymes for trimming and filling ends are also used. Then after this, the ultimate stage is to ligate the double stranded region. So now keep this thing in mind that if there is double strand break, then due to this resection process, due to this nucleases, it is also degrading some of the nucleotide bases, due to which we can lose some information. That's why this is known as the error prone repair mechanism. So because it will remove the hairpins of, or if there is some protein DNA adducts are present, so it will remove all of them, and then ultimately we will ligate it, because in this case we do not have the homologous sister chromatids, so there is no need of strand invasion or anything. We will simply join them together after the ends are cleaved. So for the ligation, the proteins which are used are XRCC4, DNA ligase 4 and XLF. They will seal the break point. And then the mechanistic features of this non-homologous end joining is that it has high flexibility. That means it is independent of processing of both the DNA ends, and it can handle diverse double strand end structures. So this is about the canonical non-homologous end joining. And then comes alternative non-homologous end joining. Now how it is different? So it is a backup pathway. If your canonical non-homologous end joining fails, or if homologous recombination fails, then this alternative NHEJ get activated. It is engaged at double strand breaks which are already processed partially by either the canonical NHEJ pathway or the homologous recombination pathway. So it's in this case the ends are ligated with the help of two enzymes, which are ligase 3 or ligase 1. So instead of ligase 4, which we have seen here, so NHEJ uses ligases 4, whereas in case of alternative NHEJ we use ligase 3 and 1. There is sensor protein which is PARP1, which competes with Ku for binding the double stranded breaks. Possible stabilizer is histone 1, for which the evidence is also available. So histone 1 is responsible for stabilizing this single stranded DNA which is generated after the nuclease activity. And then if we talk about the outcome of non-homologous end joining alternative, so this is more error prone than the canonical NHEJ because of large deletions at the junctions, and it is enhanced in G2 phase, reduced in stationary phase cells, and it may be regulated by BRCA1 phosphorylation or S988 by CHK2, which is another protein, to suppress the mutagenic alternative EJ in G2 phase. So the overall takeaway message from here is that in case of canonical NHEJ, the canonical proteins are used such as Ku7080, DNA, protein kinases. So this repair process is not that much erroneous as compared to alternate NHEJ because we are not losing much information. So this is doing short base pair resection only, and then it is joining them, it is ligating them with the help of ligase 4. But in case of alternative NHEJ, when the canonical NHEJ and HR mechanisms fails, that in this case the nucleases performs long range resection, in which long range of sequence information is getting lost, and then there are some these fragments which are formed. These swaps, they are also need to be removed when we are joining the double strand broken DNAs. So ultimately it will use the ligase 1 and ligase 3 to seal the gaps, or you can say to seal the double strand breaks. So in this case we are losing more information as compared to NHEJ. That's why it is error prone, and it is also performing the short annealing, because during resection it makes sure that 2 to 4 nucleotide stretch, if there is double strand break, then 2 to 4 nucleotide stretch is left hanging and which gets swapped off. Whereas if it is single strand break, then in that case more than 25 nucleotides can also get resected. Okay, so then this is the difference between both of them. So first binder in place of canonical NHEJ was Ku7080, whereas in case of alternative NHEJ it was PARP1. So there is competition between both of them to recognize the double strand break site. For ligation enzyme, so in NHEJ we had ligases 4, XRCC4 and XLF, whereas in case of alternative NHEJ we have ligase 1 and 3. The canonical NHEJ, it is faster repair mechanism, whereas alternative is slower. Error rate is low in case of NHEJ, whereas it is high in case of alternative NHEJ, because there is larger deletions which are taking place. And if we talk about cell cycle activity, so it takes place throughout the cell cycle process, whereas alternative NHEJ is active in all phases and it is high in G2 phase. The role of canonical NHEJ, it is primary double strand break repair in mammals, whereas this alternative is a backup when both the NHEJ and HR mechanisms fail. And these are the same steps which we have discussed previously, that how Ku7080 binds the DNA, then they will call the protein kinases to phosphorylate that particular region where the double strand break has taken place. Then Artemis and other nucleases will come which will trim the ends. Polymerase can fill the gap, and then ligation will take place with the help of XRCC4, ligase 4 and XLF, which will seal the DNA ends. Okay, now third type of recombination is site-specific recombination. So till now we have looked into the homologous recombination and non-homologous end joining. Now third type is site-specific recombination. So it means you are rearranging the DNA molecule by breaking or rejoining at specific points when you have the specific site. So with your choice you are doing this, either you are excising some segment, some gene of interest, or you are inverting it, or you are inserting something in between. So this all comes under site specific recombination. So these are the outcomes, which could be either insertion, excision or inversion. And how it is different from homologous recombination. So homologous recombination needs long homologous sequences and many proteins, because we have seen that how many proteins are involved. Whereas if we talk about site specific recombination, then it is short defined DNA sites. We exactly know which site we need to decombine or excise or whatever we want to do with the site. So that site is small, and then it is usually 1 or 2 proteins which are required to perform that site specific recombination. So now if we talk about the features of SSR, then it recognizes 2 short DNA sequences which are either same or different molecules, and a specialized recombinase which binds and rearranges the DNA. So for example, in this case, as you can see, these 2 short DNA sequences which could either be same or different, so they are recognized by some special recombinases, and the region which is present inside these sequences, that will get, it depends that what your recombinase is doing. So it can either get excised or it can get inverted, or if there are two different chromatids, then it can either get exchanged between both of them. So classical site specific recombinase, they are conservative strand exchange, no DNA synthesis or degradation, and no high energy cofactors are required. So this is one of the these are some of the points of difference from the HR. So what are the requirements for this single site specific recombination? So one is we require 2 DNA partners where we can bring about the change. Then specialized recombinase is required. This recombinase could be site recognition, cutting and joining, so it is involved in all these processes. And mechanisms that conserves the phosphodiester bond energy, because after you are exchanging the segments or you are changing it, so you're flipping it, then also you need to, because after the break you also need to construct the phosphodiester bond between that DNA fragment. So we need something which can conserve that phosphodiester bond. So examples of site specific recombination are lambda fast integration into the E. coli chromosome, resolution of co-integrates from TN3 transposition, and salmonella flagellar phase variations. So in the next slides we'll see that how this site specific recombinations takes place. The outcomes of this can be either integration, that means site on separate molecules, but the precondition is that at least one should be circular for the integration to take place. Then excision, so sites in the head to tail orientation, then excision can take place. So for example in this case this is from head to tail, so the direction is same, so in this case excision can take place. And then inversion, so sites in head to head orientation. So if we talk about these two sites, then they are facing each other, so they are like head to head, so in that case inversion takes place. Then examples of these site specific recombinases, so their function is they mediate DNA integration, excision, resolution or inversion, and these are mainly classified into two types, tyrosine types and serine type. So this type, as the name suggests, tyrosine type, that means in which tyrosine is used for catalysis, it will form Holiday junction. Whereas if we talk about serine type, then the catalysis will be done with the help of serine, and in this case 180-degree rotation mechanisms takes place. So this is the example of serine and tyrosine. So the main difference between both of them is, first of all, the molecule which is used for catalysis. So in serine recombinase serine is used, in tyrosine recombinase tyrosine is used. Second thing is, in case of tyrosine there is generation of Holiday junction, whereas in case of serine recombinase there is the rotation, so 180-degree rotation mechanism takes place. So this is about serine recombinase, and this is the example or how their superfamily is distributed. So in tyrosine recombinases also it could be bidirectional or unidirectional. In bidirectional we have Cre, FLP and R, whereas in unidirectional we have lambda, HK101 and P SAM2. In case of serine recombinase, which are also known as resolvases, so in few examples you might see syn resolvase. So syn resolvase means it is the type of serine recombinases only. They could either be small or large, and here are some of the properties. So if you're interested you can go into this, otherwise detailed information is not required. Okay, we'll talk about these two types of recombinases now, tyrosine and serine. So first of all, if we talk about tyrosine type recombinases, we know that tyrosine is the catalytic molecule. Second thing is it forms the Holiday junction like structure. So as you can see, these are two different dimers which are present, dimeric units are present. Now it will form this Holiday junction like structure here, where it will bring about the recombination now. So Holiday junction like structure will be formed, and then as you can see we started with two different dimeric, so we have two different DNA fragment, one was of purple color and another one is red in color. Now these tyrosine molecules, they will bind to this particular double stranded DNA. So now in one double stranded DNA the two tyrosine molecules have bound, and similarly in the other also two have bound. Now they are existing in the two separate dimeric forms. They will come closer and they will make a tetrameric complex, in which they will now form the Holiday junction. So in Holiday junction they will now bring about recombination. So different other proteins are also involved, same as which we discussed previously. So now in this recombinases, in these enzymes, so this will form a Holiday junction in which now they will share the fragment with each other. So both these DNA, they will share some of the strand with each other. So now in this, as you can see, previously it was completely purple, but now it shares some of the region from the red DNA as well. So both of them, they have now shared their genetic information with each other. So it is like recombination has taken place and by formation of Holiday junction. So these 2-3 things you need to keep in mind. And the examples of tyrosine type recombinases are lambda integrase, Cre and FLP. Its structure is it has conserved catalytic domain and N-terminal domains for DNA binding, and these are some of the catalytic residues. So tyrosine type recombinases, so because these are the entire protein, or you can say big enzyme. So tyrosine is a small amino acid, or is one amino acid, but this enzyme is made up of multiple amino acids. But tyrosine is the amino acid which is present in its catalytic site, which is the main amino acid which is involved. So there are some other residues also present in its catalytic site, such as lysine, arginine 1, arginine 2, histidine 2, histidine, tryptophan 3, glutamate, aspartate 1. Okay, then comes lambda phase integration or excision. So as we discussed that what does recombination means, what is site specific recombination, so in which we looked into the example of lambda phage integration. So now what happens here is, this is the bacteriophage DNA, or you can say it is a viral DNA, and this is the bacterial DNA. Now when we say site specific recombination, that means there is some site which is specifically getting recombined and no other region is getting recombined. So that's why it is called site specific recombination and not homologous recombination. Now in this case, the binding site in bacterial DNA is ATTB, whereas in the bacteriophage the binding site is ATTP. So ATT means attachment in phage and ATTB means attachment site in bacteria. Now this is the attachment site, and these are the two sites AB and XY. So for integration, lambda DNA has this ATTP site and E. coli DNA has ATTB site. Now int protein will come which will align them and recombines at 7 base pair overlap region. So this particular region, it will align both of these regions, and these enzymes, integrase, integration host factor, all these enzymes will come and they will help in the excision as well as integration. So for excision it requires int and XIS proteins. ATTL and ATTR will be synthesized. So this means attachment towards the left and attachment towards the right. So as you can see now in this O and this O, so these two O's are here, and this particular bacteriophage DNA, now it is incorporated in the bacterial DNA. So this particular stretch which is synthesized, it is known as prophage. So one DNA which was incorporated into this was circular and other one was linear, and then there is need of the attachment site which is required for this integration to take place. So this is the site which is aligned, and now entire stretch is added here. So its left side is called ATTL and right side is called ATTR, and ATTL and R are only called after the hybrid sequence has been obtained. Okay, now we'll talk about Cre and FLP, which comes under the tyrosine type recombinases only. So Cre is majorly used in the different genetic experiments in mice models. So Cre is obtained from phage P1, which targets the 34 base pair LOXP sites. So what it does is that it identifies the LOXP site and then it will specifically cleave that region. So it depends how the LOXP site is arranged. So if both the LOXP are arranged from head to tail, so the directionality is same of these both the LOX. So Cre will come and it will bind to this LOXP site and it will remove the DNA which is present here and preserve one LOXP site in the segment from where we started. So this is excision done by Cre recombinase. It can also perform inversion if both these LOXP sites, if they are facing each other, if they are head to head arranged, or you can say if they are in the opposite direction orientation. For example, this is facing forward and this is facing backward. So now whenever Cre recombinase will come, it will identify this site and it will lead to the inversion of the segment which is present in middle. But if in case if we have two different strands in which LOXP sites are present, they are facing towards the same direction, then Cre recombinase can bring about translocation. Okay, so one thing which you need to keep in mind when talking about Cre recombinase is the sites which it recognizes, the LOXP site which is up to 34 base pairs. Second recombinase is FLP recombinase from yeast to micrometer plasmid from where it is obtained. Now it targets the FRT sites. So in this figure, as you can see, this is the LPV which is the provirus and this one is the uh, DVRMCE. This is another sequence. So in both of them we have these this arrow, this represents the FRT WT site, and this uh, dark purplish arrow represents the FRT F3 site. So these FRT sites, they are recognized by this FLP. So as soon as we add FLP recombinase, now what it has done is that from this provirus it has removed the GFP-D2 till puro-TK. So the region which is present between these sites is removed and this region has been inserted or recombined here. So this is the work of FLP recombinase. So it has the sites, recognition site is FRT. Now after this we have obtained this recombined stretch of DNA. Now if we add Cre recombinase, then it will look for the LOXP site. So now it check LOXP site, it will see its direction. So if both of them are facing in the same direction, then it will cleave whatever is present in between them and preserve one of the LOXP site. So now this entire region has been deleted, one LOXP site has been preserved and the other stretch which is present is being retained. So 2 points from this slide which you need to keep in mind is that in case of Cre recombinase, the recognition site is LOXP site. In FLP recombinase, the recognition site is FRT. And if both of them are facing in the same direction, then the region which is present in between is being excised. And if they are facing in opposite directions, then region in between them will get inverted. If there are two different DNA fragments and in both of them they are facing in same direction, then recombination or translocation will take place. Okay, then similarly with the help of tyrosine type mechanisms, the recombination takes place, because these Cre and FLP recombinases, they comes under tyrosine type recombinases. And if we talk about serine type recombinases, then as we discussed, in this case in tryptophan, in tyrosine we discussed that there is the formation of holiday junction. So in the case of serine type recombinases, there is no holiday junction which is found, but there is rotation. So 180-degree rotation takes place, which is involved for the recombination to take place. So it binds the crossover site as dimers. Both of the serine molecules have attached to these two different DNA strands, double-stranded DNA strands. Now it will form the tetramer for the strand exchange to take place, double strand cleavage by the catalytic serine molecule. So as you can see, this serine molecule will perform the cleavage on both the sides. So here you can see there are breaks, or the cleavage has taken place on both the strands. And after the cleavage has taken place, then 5' phosphoserine linkage will also happen. So this protein, which is serine recombinase, it will perform 5' phosphoserine linkage. 180-degree rotation will take place of the DNA segments, and then religation will happen. So after which, as you can see, we started with one, means there are two different double-stranded DNA, one is red and other is blue. So after this, serine will bind and serine recombinase will come, so it will perform cleavage, and in this way, in the end, both of them has shared their DNA. So recombination has taken place in both of them. So the role, the examples is resolvase and invertase, and role is it resolve the plasmid dimers and separate the fused replicons. So this synthesis, this molecule is reversible or interconvertible. So it can either lead to this crossover product, or it can also convert this crossover into the known crossover product. So similarly, as I mentioned, it can resolve the protein plasmid dimers, it can act as resolvase, or it can also act as invertase. Okay, syn resolvase is also the type of serine recombinase. It is encoded by Staphylococcus aureus, which is the multi-resistance plasmid. So in S aureus we have the plasmid from where it is encoded, and this also works similarly as we discussed for the serine recombinases, that the dimer binds the DNA and then there is rotation, subunit rotation which takes place, and ultimately, for example, in this case there is invertases which are involved. So This was the when the segment has been recombined. Now from recombined segment, the it is again coming back to the known crossover products. So both ways it can work. And then there are some rest sites, which is the recognition sequences which are responsible for recognition, because every protein, they bind to the DNA at specific point or the recognition sites. So there are these specific recognition site for the resolvase subunits, and these sites could either be crossover sites for inverted, which is having the inverted repeat, or accessory sites which are either direct or inverted repeats. They have regulatory roles. So the entire application of the site specific recombination is genome engineering, because we can add the gene of interest, we can delete gene of interest, we can insert something, we can invert it. So in this way we can engineer the genome, and it is generally used for knockout and knock-in studies in mice. Mostly for this particular purpose we use the Cre-Lox system, Cre-Loxp system, and it is applicable in bacteria, plants, animals. Yeah, so this also represents the syn resolvase, that how the rotation takes place. And this is the entire summary of whatever we have discussed, the type of DNA damage and how many pathways are involved in repair. Then for double strand repair, what are the three main mechanisms, HR, NHEJ, alternative NHEJ, and steps of each of these pathways. Then we discussed the site specific recombination and how it is different from homologous recombination. Then we discussed type of recombinases, mainly tyrosine and serine, and among them also we have discussed few examples. And then we looked for the applications of these site specific recombination. So this is all about today's session, and now we'll move to the MCQs. Okay, so you have some doubt. So SSR is artificially performed or naturally occur in the organism. So this site specific recombination, this is also performed by different experimentalists, but it can naturally also it can occur. Okay, so now the first question is, what are the two broad types of DNA damage? Option A, mechanical and chemical. Option B, endogenous and exogenous. Option C, internal and external. And option D is primary and secondary. Yeah, so this is quite straightforward. We have discussed that there are two types of DNA damage, endogenous and exogenous. Now next question is, a Chi site is option A, replication hotspot. Option B, DNA damage hotspot. Option C, recombination hotspot. And option D is DNA repair hotspot. Okay, so this is also right. So the right option is option C, which is the recombination hotspot, because we discussed when we were talking about the REC-BCD in case of prokaryotes, we mentioned that it recognizes the damaged site and it will move till the Chi site is reached. So Chi site tells this REC-BCD complex that now stop your helicase and nuclease activity, and now it's the time to start the recombination process. So due to which it is also known as recombination hotspot. Yeah, and then if we talk about the replication hotspot, then replication hotspot means a site where the DNA replication starts more frequently than average. If we talk about DNA damage hotspot, then it means the region which is prone to frequent damage or breakage. If we talk about DNA repair hotspot, then it means a location in genome which is frequently targeted for repair processes. So the right answer is option C, that means recombination hotspot. Now next question is, which repair pathway is considered high fidelity? Option A, non-homologous end-joining. Option B, homologous recombination. Option C, base excision repair. And option D is mismatch repair. Yeah, you're right, because homologous recombination is known to be high fidelity because nothing is lost. It is using the information of sister chromatid to construct the lost region. And for non-homologous end joining, it is low fidelity because we might lose the stretch which is lost, which is present, means after the double strand break we do not have information that what has been deleted. Similarly, if we talk about base excision repair, then it is only accurate if there is single base pair damage. And mismatch repair, it is time consuming, expensive, and it is also for the smaller mismatches and not for double strand breaks. Now next question is what binds to the DNA ends in the initial step of double strand break repair? Option A rad51, option B RPA, option C mRN or Ku7080 heterodimer, and option D is REC-BCD. Yeah, you're right. So the right answer is mRN and Ku7080. So mRN we have seen in the case of homologous recombination and Ku7080 in case of the non-homologous end joining. Now next question is which component of REC-BCD recognizes the chi sequence or the chi site which we have seen. So option A is REC-A, option B is REC-B, option C REC-C, and option D is REC-D. Yeah, this is also right because we have seen that this REC-C, it is involved in binding the chi site. Now next question is REC-A catalyzes which of the following? Option A ATP driven homologous pairing and strand exchange of DNA molecules which are necessary for DNA recombinational repair. Option B hydrolysis of ATP in the presence of single stranded DNA. Option C ATP dependent uptake of single stranded DNA by duplex DNA and ATP dependent hybridization of homologous single stranded DNAs. And option D is all of the above. So this is in the case of prokaryotes. So similar work in case of eukaryotes is performed by rad51. Okay, so in this case the right answer is option D. That means all of these. So REC-A, it has, firstly it will bind to the single stranded region. So it requires ATP because in the figure we have seen that ATP is required to activate it, and further it will call the other REC-A to assemble on the single stranded DNA, and then it will later on send the signal for to search for the homologous single stranded DNA. So all of these works are performed by the REC-A. So answer is all of above. Now next question is in the context of site specific recombination, what is the function of tyrosine recombinase catalytic mechanism? Option A introduction of double strand breaks and rotation of cleaved DNA segments. Option B formation of a holiday junction intermediate and resolution. Option C degradation of mismatched DNA segments, and option D is extension of DNA by adding nucleotides. So this is, I guess, key difference between the tyrosine recombinase and the serine recombinase. Okay, now let's see. So the right answer is option B, which is the formation of holiday junction intermediates and resolution. So if we talk about option A, then they mentioned introduction of double strand breaks and rotation of cleaved DNA segments. So rotation takes place in the serine recombinases and not in tyrosine recombinases. And if we talk about holiday junction intermediates, then yes, in case of tyrosine recombinase, we have seen that how the holiday junction intermediates are formed. Similarly, C option says the degradation of mismatched DNA segments. So degradation, so there is no mismatched DNA is present in case of tyrosine recombinase, and this is observed in the case of mismatch repair mechanism. Then D option was extension of DNA by adding nucleotides. So this extension, this is the work of DNA polymerases and not the recombinases. Okay, now let's move to next question. So which site specific recombinase is known for its use in genetic engineering to induce the recombination at specific sites in the genome? Option A Cre recombinase, option B REC-A, option C RUVC, and option D is REC-G. Okay, so yeah, option A is right, which is Cre recombinase. So we have seen that this is most commonly used. So the site which it recognizes is the LOXP site, and it depends that in which orientation LOXP site is present. So in that way, Cre recombinase decides whether to excise the DNA, to invert the DNA, or to translocate that particular stretch of DNA. So it is majorly used. Whereas REC-A, so we discussed during homologous recombination in prokaryotes that the REC-A it is generally used, and we have seen its work, that how it binds to single-stranded DNA involved in the homologous strand identification and all those processes. For roof C, so RUVC is the holiday junction resolvase in homologous recombination. And rec G, so this is the helicase which unwinds the DNA during recombination and repair, and it is also not used in the case of site-specific recombination. Okay, so let's look at the next question. So which protein is essential for excision of the phage DNA from the bacterial DNA in the lambda phage system? Option A, int protein. Option B, XIS protein. Option C, both int and XIS proteins. And option D is Cre recombinases. Okay, now let's see. So in case of excision, we require both the int as well as XIS IHF proteins. Int protein is the integrase protein, which is the enzyme required for inserting the lambda phage DNA into the host genome during the lysogeny, and also participates in excision, but it cannot excise on its own. So for excision we require both int and XIS. So XIS protein is also known as excisionase protein, which assists in excision by bending the DNA and helping int to recognize the recombination sites for removal of phage DNA. But see here also it cannot act alone, so both of them act in a complex. Both of them are required for excision, so that's why option C was right. And Cre recombinase, so we have seen in case of Cre-Loxp system, Cre recombinase is used, and not in case of this lambda phage system. So the right answer is both int and XIS IHF proteins. Next question is, what triggers the formation of the tetrameric species of syn resolvase? So syn resolvase, the other name is serine recombinases. Option A, DNA polymerase activity. Option B, accessory proteins, high local concentration or mutations. Option C, RNA interference. And option D is spontaneous DNA damage. Okay, so it's alright if you do not have any idea. So the right answer is option B, which is accessory proteins, high local concentration and mutations. So all these things, they trigger the syn resolvase, and it converts the syn resolvase, which actually exists as dimer, and convert it into the tetrameric complex, which is the active form of recombination at the rest site or the recognition site. If we talk about DNA polymerase activity, so the syn resolvase, as it is recombinase, so it do not have polymerase activity, and it is also not involved in RNA interference, and similarly not in spontaneous DNA damage. So because it is serine recombinase, so in this case there will be subunit rotation. This is the same figure which we have seen in the serine recombinases. Okay, so next question is, which type of DNA damage is most commonly induced by ionizing radiations? So option A is single strand breaks, option B is double strand breaks, option C is cyclobutane pyrimidine dimers, and option D is base mismatches. Okay, now let's see. So right answer is option B, but you are right. The ionizing radiations can also lead to single strand breaks, but these single strand breaks, they are repaired more frequently and easily. So the most significantly observed DNA damage by ionizing radiation is double strand breaks, because these ionizing radiation, they generate the reactive oxygen species, which can break both the strands of DNA, and due to which this double strand breaks, they are more frequently observed, and they are also dangerous also. If we talk about cyclobutane pyrimidine dimers, then they are generated by UV radiations, and base mismatches, so they result from the replication errors and not directly by the ionizing radiations. So the right answer is option B. Next question is, which protein is directly responsible for loading RAD51 onto the single stranded DNA during homologous recombination in eukaryotes? Option A, BRCA1, option B, RPA, option C, BRCA2, and option D is KU7080. Yeah, so you are right. because we have seen that BRCA2 and PAL2, both of them, they bring the RAD51 and help it to replace the RPA. So if we talk about BRCA1, so it functions in the early stages of homologous recombination by promoting DNA end resection and interacting with BRCA2, but it is not involved in loading RAD51. Then if we talk about RPA, then we have also discussed that whenever the resection is taking place, then the single-stranded DNA is unstable, so for which we require RPA to bind it and to protect it from the nucleases, and ultimately it get replaced by RAD51 and BRCA, which is the right option, BRCA2, which is responsible for loading the RAD51 onto single-stranded DNA and replace the RPA. Then if we talk about KU7080, so as in case of homologous recombination we have mRNA, similarly in case of non-homologous end joining we have KU7080, which recognizes the site of double strand break. So next question is, in base excision repair, which enzyme removes the damaged base? So option A is AP endonuclease, option B is DNA polymerase beta, option C is DNA glycosylase, and option D is DNA ligase 3. Yeah, so this we have discussed in the previous session. So the enzyme which is involved in the base excision repair is DNA glycosylase. So if this is a DNA damage, then glycosylase will come, it will remove that base and it will generate the AP site, which could either be a purinic site or a pyrimidinic site. AP endonuclease will come, it will cleave it, and then if there is short patch being deleted by AP endonuclease, then it will go from here. If there is long patch deletion, then it will go from here. DNA polymerases will come, they will synthesize the strand. Endonuclease will come, which will degrade the extra strand if available, and DNA ligase will seal the gaps and create the phosphodiester bond between the nicks which are created. It will seal the DNA. Okay, so next question is, which recombination repair protein in bacteria promotes the branch migration of holiday junctions? Option A REC-A, option B REC-G, option C RUV-A, and option D is RUV-B. Okay, so now this is a bit tricky because we have talked about REC-A and we have not talked about RUV-A and RUV-B. So in this case, RUV-B is mainly involved. So REC-8 catalyzes the homologous pairing and strand exchange during recombination, but it does not perform branch migration. So this we have talked, that homologous pairing and strand exchange, but this branch migration, it is the different process, so which has been performed by RUV-B. So let's talk about REC-G. So REC-G is a helicase which can promote branch migration independently, but in this they have also talked about holiday junction. So in main holiday junction resolution pathway, it's not the primarily motor protein used. So in this way, REC-G is also not right. If we talk about RUV-A, then it recognizes and binds to holiday junction, forming complex with RUV-B, but it does not power the migration. So in this, as you can see, this middle one, orangish yellow, this represents the RUV-A, whereas this reddish part, it represents the RUV-B hexamer. So although this RUV-A is responsible for binding and recognizing the holiday junctions, but for the branch migration, the power is being provided by RUV-B hexamer. So RUV-B is the ATP dependent helicases which works with RUV-A to drive the branch migration of holiday junctions, and it moves the crossover points along the DNA. So for example, if in another time if option comes where RUV-A and RUV-B both are mentioned together, then that will be the right option. But in this case, among both of them, main is the RUV-B. But it doesn't mean that RUV-A is not playing role. Both RUV-A and B together, they are involved in the branch migration. Yeah, so keep these two proteins also in your mind. Now next question is, in eukaryotic DNA double strand break repair, which complex is the first to bind the broken DNA ends in non-homologous end joining? Option A mRNA complex, option B KU7080, option C ligase 4 and XRCC4, and option D is DNA PKCS. Okay, so Yes, you are right, because we have discussed that in non-homologous and joining, the protein complex which recognize the double strand break is Ku7080. mRNA is in homologous recombination, ligase 4-XRCC4, it is involved during the ligation process, and DNA PKCS, it comes after Ku7080 to phosphorylate the site and further bring other uh, proteins which will further bind and perform the degradation. Okay, so next question is, which protein complex is the primary sensor of double strand breaks in eukaryotic homologous recombination? Option A Ku7080, option B mRNA complex, option C BRCA1 PAL-P2 complex, and option D is DNA PKCS. So these questions are coming repeatedly because we've looked into these many times. So because by the end of this session, now all these names will be on your tips. Okay, so you are right. So the right answer is option B, mRNA complex, as you can see here. So in homologous recombination, there is mRNA complex. Next question is, in homologous recombination, which protein replaces RPA on single-stranded DNA to form the presynaptic filament? Option A RAD54, option B RAD51, option C REC-A, and option D is BRCA1. Yeah, so you're right, because we have discussed that RAD51 is the one which will replace the RPA, and this replacement is further helped by BRCA2 and PAB-B2. RAD54, so if you remember when we were talking about accessory proteins, then we discussed about RAD54 that it interacts with the RAD51 to remodel chromatin and stabilize the D loops, because this RAD51 lead to the D loop synthesis. So RAD54 is accessory protein which will help the RAD51 to work properly, whereas REC-A, so we have read about it in the case of bacterial homologs, that in bacteria we have the REC-A in place of RAD51 which does the similar work as compared to RAD51. And BRCA1, so we discussed that it is involved in the initial steps which assist in RAD51 loading indirectly and not directly involved in this RAD51 loading. Now let's see the next question. So next question is, which catalytic residue is responsible for DNA cleavage in tyrosine type site-specific recombinases? Option A serine, option B tyrosine, option C lysine, and option D is histidine. Okay, so when we were talking about Cre, FLP recombinases, so they come under tyrosine type site-specific recombinases, and the work of site-specific recombinases is to identify one particular site, bind to it, and then cleave in between whatever is present, so cleave it. So that cleavage is brought about by some of the amino acid which is present in those recombinases. So when we say tyrosine recombinases, so they have tyrosine for that cleavage. This doesn't mean that they only have tyrosine, they have other residues in the catalytic site, but the main residue which is involved is tyrosine in case of tyrosine type site-specific recombinases. And in serine type site-specific recombinases, the catalytic molecule will be serine. Next question is, the catalytic mechanism of serine type recombinases includes option A formation of a holiday junction intermediate, option B 180-degree rotation and rejoining of cleaved DNA, option C use of ATP hydrolysis for the strand separation, and option D is DNA degradation before integration. So now in this we are talking about serine type recombinases and not about tyrosine type recombinases. So the right answer will be option B, which is 180-degree rotation and rejoining of cleaved DNA, because this holiday junction, it is formed in tyrosine type recombinases and not in serine type recombinases. Yeah, so keep this thing in mind that in serine type recombinases there will be 180-degree rotation, and then this cleaved DNA, it will be recombined. So next question is, in bacteriophage lambda integration, the bacterial attachment site is called ATTP, ATTB, ATTL and ATTR. So keep this thing in mind that they are talking about bacterial attachment and then bacteriophage. So bacteriophage is like virus and bacteria is bacteria. So to remember the attachment site, uh, keep focus on the word bacteria. So in bacteria we have B. In bacteriophage we cannot use B because B is specific for bacteria, so we will use P. So in case of bacterial attachment site it will be ATTB. So as here we can see, this is ATTB site and this is ATTP site. Both of them, they are aligned to one another, and then other proteins, INT, RDF, all these proteins will come which will help to recombine both the fragments. So ultimately prophage will be constructed where we will be having this phage DNA in between these sites. So another thing which I want to mention is about ATTL and ATTR. So ATTL means in the hybrid, when the hybrid is synthesized, so this is the hybrid attachment site formed after integration. So this shows the left junction between the bacterial and phage DNA. So this is the left junction, ATTR is the right junction. So in this case the left part is of bacteria, bacterial part is towards the left side, and this bacterial part is towards the right side. So this is joining of both of these phage DNA and bacterial genome together. Now next question is, which domain in Rec A contains the ATP binding site? Option A, N-terminal domain. Option B, core ATPase domain. Option C, C-terminal domain. And option D is flexible tail region. So you need to tell that which domain contains the ATP binding site. So you can guess from these names that where the ATP site could be present. Yeah, so means this guess is right. So option B is right, which is core ATPase domain. So when we looked into the figure, we know that there is some N-terminal domain as well as C-terminal domain. Both of them do not bind to ATP. They have central site where the ATP binds, and this site is present in this domain which is known as core ATPase domain. So core means it is present in the internal site, it is not externally present. So this is the core site, core ATPase domain, where the ATP can bind. And if we talk about the flexible tail region, so it modulates the activity and interactions, but it has no role in ATP binding. If we talk about this C-terminal domain, then it is involved in double-strand DNA gateway function and regulation of activity, but it is also not involved in the ATP binding. So you can see this yellowish part represents the C-terminal domain, and it has double-strand DNA interactions. So this interacts with the double-strand DNA. So if you remember that during the homologous recombination, when Rec BCD binds, and then whenever Rec C identify the chi site, then it says stop the nuclease activity, it will call Rec A. Rec A will bind to the DNA. So this particular region, it is responsible for that interaction between the double-stranded DNA. Whereas this orangish part, which is site 1, it will bind the single-stranded DNA. So if we talk about N-terminal domain, then this is also involved in the double-stranded DNA interactions. Now next question is, in the lambda phage excision process, which protein works together with integrase? Option A, HIN, option B, FIS, option C, XIS, and option D is HU. So yeah, you are right. So the right answer is option C, which is XIS. We have already seen that during integration, int IHF is used, but during excision, we require both int and XIS along with IHF. They together are responsible for the excision. So let's also discuss about other options. So this HIN, this is a site-specific recombinase from Salmonella, and it is used for the DNA inversion. So it is not involved in the lambda phage excision. Whereas if we talk about FIS, so this is a bacterial nucleotide associated protein which regulates the transcription and recombination, but it is not for the excision partner in the lambda phage. If we talk about XIS, then yes, it along with the int and IHF, it is involved in excision. If we talk about HU, then it is the bacterial DNA binding protein that can assist in integration and excision indirectly, but it is not the main partner. So right option is option C. Now next question is, which non-homologous end joining ligase complex seals the DNA ends after processing? Option A ligase 1 XRCC1, option B ligase 4 XRCC4 and XLF, option C ligase 3 and PARP1, and option D is ligase 2 and KU70. So in non-homologous end joining, which is the canonical one, so in which ligases are used? Yeah, so you are right. So the main difference between the non-homologous end joining and the alternative end joining or alternative non-homologous end joining is in the ligases only. So in NHEJ we use ligase 4 XRCC4 and XLF, whereas in the case of alternative we have ligase 1 and 3 along with PARP1. So instead of KU7080, PARP1 competes with them. Yeah, and in also C for HR also. In HR we, okay, so in this it's not mentioned. So in HR we have, I guess, ligase 1. There are some other type of ligases which are responsible for the ligation process. So next question is, what is the primary role of the BLM helicase in homologous recombination? Option A promotes crossovers in mitotic cells. Option B suppresses crossovers and resolves the double holiday junctions. Option C loads Rad51 onto single-stranded DNA, and option D is recognizes the chi sequences. So we discussed about the BLM helicases when we were talking about the double holiday junction, and we were discussing that whether the joint molecule will lead to the crossovers or it will lead to the non-crossovers. So in that we discussed that where the BLM helicase was used. So the right answer is option B, which is suppresses crossovers and resolves the double holiday junctions. Okay, so the first option was promotes crossovers in mitotic cells. So this is completely wrong because it actually suppresses the crossovers. The option B is suppresses crossovers and resolves the double holiday junctions. So it works along with the top 3A and RMI 1/2 complex to dissolve the double holiday junctions without crossovers. So if you remember that figure, so in that figure we discussed that if there is nick and ligation, then the double holiday junction will give rise to the crossover products. But if BLM helicase along with these two protein is used, then it will generate the non-crossover molecules. Yeah, the third option was loads Rad51 onto single strand DNA. So this is performed by BRCA2 and PALB2 and not by BLM. And the recognition of chi sequence, so this is done by REC in bacteria and not in by BLM in eukaryotes. Okay, so next question is, which serine recombinase is encoded by Staphylococcus aureus multi-resistance plasmids? Option A HIN, option B SYN, option C FLP, and option D is CRE. Okay, so any guesses? Yeah. So we discussed about SYN resolvase. So SYN resolvase comes under serine recombinase only. So if we talk about HIN, so it is obtained from Salmonella, and this is the example of tyrosine recombinase which is involved in DNA inversion for flagellar phase variations. Whereas if we talk about SYN resolvase or SYN, this is the example of serine recombinase which is found in Staphylococcus aureus multi-resistance plasmids, and it mediates site-specific recombination. If we talk about FLP, then it is also the example of tyrosine recombinase, and it is obtained from Saccharomyces cerevisiae, which is the yeast, and this is used in genetic engineering for FRT site recombination. So the example of CRE and FLP, where we discussed that this FLP, it recognizes the FRT site. Similarly, if we talk about CRE, then this is also the tyrosine recombinase type, and this is obtained from bacteriophage P1, and it recognizes the LOXP site, and it brings about recombination, and it depends that both the LOXP sites in which orientation they are available. So based on that, it will decide whether to perform inversion, insertion, or excision or translocation. Okay, so now next question is, Which type of recombinase does not require host encoded accessory proteins or specific DNA structures for activity? Option A, lambda integrase. Option B, Hin invertase. Option C, Cre recombinase. And option D is Gin recombinase. It's all right. So the right answer is option C, which is the Cre recombinase. And this particular question, it also tells you the advantage of using Cre recombinase because it does not require any accessory proteins, due to which this becomes more favorable to use. So if we talk about lambda integrase, then it requires host encoded accessory proteins such as IHF, FIS, and specific DNA structures at the attachment sites. If we talk about Hin invertase, then it also requires the accessory proteins such as FIS and specific DNA enhancers for their activity. But the Cre recombinase, it functions independently and it only requires the LOXP sites and do not require any host proteins or special DNA topology. Now the last option was Gin recombinase. So it requires accessory proteins and the recombination enhancer sequence for its activity. So the right answer was option C, Cre recombinase. And similarly, the orientation of these recognition site will decide whether the outcome will be inversion, deletion or translocation. Next question is, in alternative end joining, which is AEJ, which ligases can complete the DNA end ligation? Option A, ligase 1 and ligase 4. Option B, ligase 3 and ligase 1. Option C, ligase 4 and ligase 3. And option D is ligase 2 and ligase 4. So the right answer is option B, ligase 1 and 3. In NHEJ, we talked about ligase 4 with XRCC and other protein. So keep this thing in mind that in non-homologous end joining, we have ligase 4, which will not be used in alternative end joining. So one major difference between both of them is of the ligases. So in alternative end joining, we have ligase 1 and 3. So whenever there is double strand break, then in place of Ku7080, which were used in the non-homologous end joining, here we have PARP1 and histone 1, which will stabilize the single stranded DNA which is generated. Then other proteins, CTIP, mRN, they are also available. So they will come. Polymerase theta they have mentioned for the extension. So polymerase theta is used here, and then for the ligation, ligase 1 and 3 along with XRCC1, they are involved for ligation. Yeah, so in all other options, ligase 4 was present. So that's why this is the right option. Now next question is, which accessory proteins are required by lambda integrase for excision of the prophage DNA? So this question, I guess this is now coming for the third time. So option A is FIS and HU, option B XIS and IHF, option C is Cre and FLP, and option D is BRCA1 and 2. So yes, the B option is right. XIS, IHF along with integrase, so all of them together are responsible for the excision, whereas FIS and HU, they are the DNA binding proteins and not for excision, whereas Cre and FLP also we have discussed, they are tyrosine type recombinases which are not used in this lambda phage integration, and similarly BRCA1 and 2, they are also involved in the homologous recombination, where BRCA1 was used initially, whereas BRCA2 with BAB2 was involved in the loading of RAD51. So yes, right answer is option B. Now next question is, in BCD processing, what change occurs after the recognition of a Chi site? So option A, nuclease activity stops completely. Option B, degradation of 3' tail is attenuated and 5' tail degradation increases. Option C, DNA ends are ligated immediately, and option D is Reg A is removed from the DNA. Okay, so somewhat what we discussed is that after the Reg C, it identifies the Chi site, it stops the nuclease activity, but still the degradation of 3' tail is stopped, but 5' tail degradation increases. So this is the thing which I forgot to mention. So according to studies, they have mentioned that the nuclease activity stopped, but what nuclease activity? So degradation of 3' tail which was taking place, that stops, but the 5' tail degradation, it increases. So keep this thing in mind, that when we say nuclease activity, so basically previously the nuclease activity which we were talking about was the degradation of 3' tail, because from the 3' ends in both the double strand breaks the degradation was taking place, so that thing stops, but at the same time the 5' tail degradation was increasing. So due to which this nuclease activity stops completely is not the right answer. And then option C is DNA ends are ligated immediately, so this is also not right. And then rec A is removed from the DNA. So this rec A, it loads after the rec BCD reaches the chi site, so this is also not right. So that's why the right option is option B. Now the last question is, which type of site specific recombination intermediate is the characteristic of tyrosine recombinases? So option A is hairpin DNA, option B is holiday junction, option C is D loop, and option D is nicked duplex DNA. So in this you have to tell which is the intermediate characteristic of tyrosine recombinases. So this also, when we were talking about the tyrosine versus the serine recombinases, so this we discussed. Yeah, so this is right, holiday junction. So if we talk about hairpin DNA, so this is the characteristic of serine recombinases, and the example of serine recombinases are HIN, GIN or VDJ recombination. And when we talk about tyrosine recombinases, then yes, holiday junction is the characteristic feature of that, it forms holiday junction. If we talk about D loop, then D loop is formed during the homologous recombination when the RAD51 invades the strand. And nicked duplex DNA, so it could be transient form in various repair processes, but it is not the hallmark of tyrosine recombinases. So the right answer is option B, holiday junction. Yeah, so these are all the questions about today's session. So I hope by the end of this session you will be able to remember all the names, all the proteins which we have discussed for all these repair pathways. So if you have any doubt, then you can ask, otherwise I'll end the session. And in the session, if you are not able to attend them live, then from the YouTube videos also you can comment below if you have any questions. So okay, if you don't have any doubts, then I'll end the session, and thank you for joining. Okay, so you are confused between tyrosine and serine type recombinases, so means they are not that much difficult. So in case of tyrosine recombinases, keep one thing in mind, that tyrosine recombinases, they use tyrosine as a catalytic molecule. Both of them, they bind as a dimer to single double stranded DNA, and then both of these dimers, they will join together to form a tetramer. So in case of tyrosine type recombinases, they will construct a holiday junction. When both the double stranded molecules, they will come closer, then they will form a holiday junction. Whereas in case of serine type structure, they will form the hairpin loop like structure, in which there will be rotation, and after the rotation the recombination will take place. So these are the 2 points which distinguish between the tyrosine and serine type recombinases. And if you still have any doubts about these tyrosine and serine type recombinases, then yeah, so I will add the details, or I'll suggest some reading means documents if I have. So yeah, this is about today's session, so I'll end it. Thank you very much for joining, and see you in the next week.